Carolina. She then went on to receive her PhD in molecular genetics from the Medical College of Virginia, and subsequently, she went to Vanderbilt University Medical Center to complete her ABMGG fellowship in clinical molecular genetics. Currently, Dr. Vnencak-Jones serves as professor of 3 different departments: pathology, microbiology and immunology, and pediatrics. And she's also the director of the Molecular Diagnostic Laboratory for Vanderbilt University Medical Center. Today, Dr. Vnencak-Jones is going to speak to us on precision medicine for the right drug at the right dose on the first time.
Okay, good morning. It's great to be back at a SERGG meeting. It's been a few years since I've been at one. And I'd like to first begin by thanking the organizers for inviting me to share with you our experience with the Pharmacogenetics Initiative at Vanderbilt. I have no disclosures to report. I do want to show you, this is the This is the Vanderbilt Main Medical Center campus. It is adjacent to the Vanderbilt University. We're about 2 miles from downtown Nashville and the Bridgestone Arena, which the Nashville Predators were in the Stanley Cup Finals. We didn't quite go all the way, but I think we put up a good fight. So the objectives today, at the end of my talk, you will be able to differentiate between various allele types and understand their associated clinical phenotypes. You will recognize, I hope, that pharmacogenetic testing enables precision medicine. You will understand what we call drug genome interactions for some commonly prescribed drugs. And you'll be able to evaluate the utility of pharmacogenetic testing in routine patient care and the use of bioinformatics to store the data and facilitate decision support for appropriate drug therapy. So before I actually go into the nitty-gritty of our initiative, we're going to talk a few minutes about some basics so we're all on the same page. I don't know how many of y'all deal with pharmacogenetics in your settings, but we're going to do some basics so that we can all be at the same page. So what is pharmacogenetics? Well, it's the influence of genetic variation in modulating the response to pharmacologic agents. And when we think about that, we think about pharmacokinetics, which is the efficacy of the drug. How well does that drug work within that person? And pharmacodynamics, which is the safety of the drug. And the important thing about pharmacogenetics is that it enables genotype-driven drug therapy. So why is this an important topic? Well, pharmacologic agents can lead to adverse drug reactions. And that's a pretty big deal. About $30 billion in healthcare resources annually are used because of adverse drug reactions. It's the 4th cause of death among hospitalized patients. So when we think of an adverse drug reaction, what is the cause? Well, you can have it caused by drug-drug interactions. So you have multiple drugs that share the same metabolizing enzymes. So there's like a competition. You can have drug-disease interactions because that can be dependent on the functional status of the host. If the host has liver disease, they're not going to metabolize the drug as well. If they have renal disease, they're not going to excrete the drug as well. If they have heart disease, they're not going to circulate the drug as well. So all of these can play a role in how well the efficacy and the safety of the drug is within the individual. You can also have drug-food interactions where you can have components within certain foods can inhibit certain enzymes, which are used to metabolize drugs. And you can have what we call drug-gene interactions, where the genotype encodes the proteins, and these proteins are what's involved with the absorption, distribution, metabolism, or excretion of the drug. So all of these different things can cause an adverse drug reaction. And they're becoming increasingly problematic because there's more and more drugs on the market, with $320 billion in prescription drugs annually. Because you have more drugs, you have the chance of more drug-drug combinations because more people are prescribed more drugs. And the important thing is that a significant percentage of these adverse drug reactions could be preventable if you had the genotype information. So here's another important part. Why is pharmacogenetics not part of routine patient care? Well, there's limited guidelines for these drug genotype interactions. There are two big groups that are working on it. The Clinical Pharmacogenetics Implementation Consortium, commonly called CPIC, and then you also have the FDA working on it. So these are the two big groups that are actually working on and setting guidelines for various drugs and genotypes. And there's multiple studies out there in the literature, as well as one that we did within our own institution, that says about 10% or less of healthcare providers actually feel knowledgeable in this area. So it's very difficult for them to order these assays if they don't know what to do with the information. So that's a big problem. There's also limited evidence of cost effectiveness, like really what would, should we be doing this testing? Is it going to be cost effective to get these results? And how will they be implemented in patient care? And will that save money? There's also limited evidence of clinical utility. And then you have the problem of delayed treatment while you wait for the results to come back. There's limited availability of alternative therapy. So if somebody can't use this drug, what drug should you give them? And then there's lack of informatic tools to store and deliver these results. So the next thing I want to talk about is nomenclature for pharmacogenetics, because it's a little bit unusual. It doesn't fit the typical genetics nomenclature that we're all familiar with. The sequence variants are designated as star alleles. A star 1 allele is designated as normal, wild type, fully functional, and a normal allele is called an extensive metabolizer. So that would be the phenotype of a star 1. They would be called an extensive metabolizer for that drug. Star alleles are identified by a defining variant, but actually they represent a unique haplotype of many linked SNPs. Within each star allele, there are additional variations that are linked in cis to the defining SNP, and these represent subfamilies. So star 2A, star 2B, and star 2C. So this concept shows that if the star 2 allele is a G to T substitution, a star 2A allele has both the star 2 allele as well as a unique SNP in cis. A star 2B would have the star 2 allele as well as a unique other SNP. But remember that all the star 2 alleles, while they have this SNP, which is unique to them, there are many SNPs within that whole gene. But this is the one that would define it as star 2. And then variants are numbered in the order in which they were identified and not their sequential order within the gene. So all this can make it kind of confusing. So this is just an example of one gene to illustrate what I'm talking about in terms of many different polymorphic sites within this gene that convey a clinical significance. So when you see CYP2C19, what that actually means is cytochrome P450, so it's a member of the P450 set of proteins. It is family 2, subfamily C, polypeptide 19. And within this gene, you see I have multiple star alleles highlighted here, but these variants represent missense mutation, premature truncation, splicing, promoter, and insertions. So there's lots of different types of variants in each one of these genes that metabolizes these various drugs. So the last piece of basic information that I want everyone to understand is that there is a phenotype associated with each of these genotypes. So the genetic polymorphisms result in variations in the drug metabolizing enzymes. So for example, if your genotype is 2 functional genes, normal, wild type, you're called an extensive metabolizer. If you have 2 or more functional genes, so you have a copy number variant, or you have a variant in the promoter region that makes a lot of the transcript, so you're going to make more of that protein, you're called an ultra-rapid metabolizer. If you have 1 functional and 1 defective variant, or 2 partially functional genes, such as a wild type and a stop codon, or 2 amino acid substitutions, you would be called an intermediate metabolizer. And lastly, if you have null or low functional genes, frame shifts, splice variants, stop codons, you're going to be considered a poor metabolizer. So your phenotype, based on your genotype, refers to metabolizer status. Okay, so when somebody gets a new diagnosis and they're prescribed a drug, generally what happens is they have an empiric dose given, a one-size-fits-all. That one-size-fits-all was based on clinical trials, usually about 2,000 to 3,000 patients studied. And there's a risk for an adverse drug reaction because 2,000 to 3,000 people do not represent the whole world. So there's many different genotypes that weren't found in those people. So the people now who are taking this approved drug could now actually have an ADR. Then it's possible that you are at an institution or a doctor's office where they order a genetic test and you're given the drug, perhaps the one-size-fits-all, but within a week or so, you're contacted and your drug is changed or the dose of the drug is changed. And so all this is happening real time and we hope that an ADR has been avoided. But ultimately, the goal would be to have this genotyping data on hand and ready to go so that when you need a drug, we know what's in your files, and you would be prescribed the correct medicine at the right dose at the first time. So with this goal in mind, Vanderbilt launched their PREDICT initiative. And it was launched in 2010, and the long-term goal was to establish a framework and infrastructure for real-time and preemptive incorporation of genomic data into the EMR. So before we launched it, there were several things that were done ahead of time. First of all, there had to be a commitment from a multidisciplinary team at the institution. So we had clinicians, ethicists, informaticians, and all different kinds of people involved in this, because there's lots of moving parts. We had to develop educational material for the providers, because remember, many of them don't understand pharmacogenetics and what to do with that information. We had patient focus groups, and they all said that PREDICT, or our preemptive genotyping, should be done. We had online surveys through our My Health at Vanderbilt, where people could answer questions. Would you like to have your DNA tested for certain drug metabolizing enzymes and have that stored in your medical record? And the vast majority said yes. So then we had PREDICT information added to the consent to treat form so that it became an automatic testing that was done in the preemptive setting. Then there was a lot of review of literature by and professional societies and the FDA so that we could decide what would be the drug genome interactions. In other words, what actions should be taken when a certain genotype is found. And then we had to identify the assay that we were going to use and validate it. So the next piece of material we had to determine was what were the genes and the drugs that we were going to study and report. And to do that, this was determined by our Pharmacy and Therapeutics Committee. And when they did this, they took many things under consideration, including the significance of impact of a negative outcome. Was there compelling and consistent body of evidence to support this particular drug genome interaction? How often was that drug prescribed at Vanderbilt? Was the gene testing performance accurate and reliable? Was there an availability of an alternative therapy? So if you find that somebody is a poor metabolizer for a commonly prescribed drug, what are you going to do then? Is there an alternative therapy for them? Could it work into our workflow? And what was any regulatory guidance concerning this? So the first one that we launched was clopidogrel, which is also called Plavix. It is often used for people who've had acute coronary syndrome, a recent myocardial infarction or stroke, peripheral arterial disease, and mostly also people who have stent placements. It is generally given at an initial dose of 300 milligrams, followed by a daily dose of 75 milligrams. And at the time, in 2010, there was, and there still is, but it started in 2010, a black box warning by the FDA saying that people who are poor metabolizers of this drug are at an increased risk for thrombosis. It is widely prescribed. It's an antiplatelet therapy with $6.5 billion a year in prescriptions. So how does it work? So first of all, clopidogrel is a prodrug, so it must be converted to the active form. It's converted to the active form primarily through the gene I just talked about, CYP2C19. If it's not converted to the active form, then it remains in a prodrug state and it's not usable by the individual. Okay? What it does, it actually binds irreversibly to platelets, to the receptor molecule on the platelets. And what it does is it prevents the platelets from aggregating. So it prevents a thrombosis event. So what it means, according to the FDA guidelines, is that if you have reduced functioning alleles, if you are a poor metabolizer, you will have significantly lower levels, depending on what the variant. You may have no levels of the active metabolite because you were unable to convert it from the prodrug to the active form. If you have low levels of the active metabolite, then you have diminished platelet inhibition and a higher rate of platelet aggregation and a higher rate of a stent thrombosis, for example. So this is pretty significant. So just a quick little laboratory review of how we actually are doing the test now, so you can kind of start putting all of the pieces together. We use a TACMAN assay. So we do it in 96 well pre-made plates. So everything we need for the assay basically comes, all the primers and everything are in the plates. It's a TACMAN assay, and in this case, if you have an A allele, then for one particular variant, if A is wild type, let's just say, and G is mutant, we're going to test at that particular variant, do you have the A or the G? So the way you do that, it's a TACMAN assay. The PCR product is made and 2 probes are added. One is specific for the A and one is specific for the G. If the A is present, then you will generate the fluorochrome Vic over here. It will be given off. If you have G, then you will have FAM generated. So someone who is GG would have a lot of FAM. Someone who is AA would have a lot of Vic. And that's how you genotype the people. And this is just an example of a cluster plot. So these would be people who are GGs, an AG, and an AA. Okay, so that's just a brief thing on how we're doing the test now. Okay, so now we can put it all together. So for CYP2C19, which is used to metabolize clopidogrel, we test for 6 different variants. These variants are associated with poor metabolizer or ultra metabolizer state. So we would generate a report that said the patient in this case has a *2 in one allele and a *3 in another allele. So that's actually the raw data. So the person is a compound het with a *2, *3. So that's what the lab report is saying. This *2, *3 generates a drug genome interaction that says, for clopidogrel, this person is a poor metabolizer. They have reduced antiplatelet effect with 2 loss of function alleles and no gain of function alleles. And then it refers everybody to my drug genome, which we're going to talk about in a few minutes. We also have a pediatric DGI, and in this case, there is not current evidence to support the use of genetic data for guiding in the pediatric population. But we do report that they have a *2 and *3, which are poor metabolizers. What that actually means in the patient population, there isn't a lot of data, and this is all data-driven. So then, besides seeing the lab report, besides getting the DGI, when the clinician goes to order the drug, we provide we provide decision support. So we tell the clinician when they go to order online the drug that this person is a poor metabolizer of clopidogrel. And so you might want to prescribe another drug that does not require CYP2C19 to metabolize it. Therefore, you would, by giving them this other drug, then you would alleviate the fact that the person is going to get no benefits from clopidogrel. And so we actually tell them this is what you should do, and then they can read about it. We tell them again, this is why we're suggesting that you do this, and then they can opt to either choose the drug that we suggest or continue with clopidogrel. So that's the decision support that's all linked together. So you may think that these are, in genetics, we oftentimes think of rare variants, rare genotypes. You may think that these are rare. These are not rare. In our patient population, only about 70% have the star 1 alleles, normal metabolizers. 21% you would recommend alternative therapy because they have a poor metabolizing allele. 3% are homozygous for poor metabolizing of this drug. They will get zero benefits from Plavix. And so basically, we have almost 24% of our population who really are not getting the complete efficacy of that drug. And 5% would be homozygous for our ultra-rapid metabolizers. They would still get clopidogrel. You would not change the therapy based on that. Okay, so we began with CYP2C19, which I've mentioned is an antiplatelet medication with about 24% of our population requiring a different drug to be used. After that, we went to simvastatin. Simvastatin is a very commonly prescribed cholesterol-lowering drug. This is a transport molecule. You actually need this variant, this protein, to bring the drug into the liver to be metabolized. And if you don't do that, then it circulates in the body, and when it gets into muscles, because it's not going into liver, it actually causes a lot of muscle pain. So some people who take simvastatin may not want to take it anymore because they're getting muscle pain from it. And the best thing to do is just prescribe them a different statin, because not all statins are created equal, and not all of them require SLCO1B1 to bring it into the liver. So again, in our population, in about 24% of our population, we suggest lowering the drug amount, or in 2%, give them a completely different drug. We also then launched warfarin. Warfarin, everyone I'm sure is familiar with. It is an anticoagulant. And in our population, 77% of our population should have used a lower dose than the empiric dose. Then we launched TPMT, which is a methyltransferase. It's mostly used to treat people who have leukemia. And the reason to do it is because it's has myelotoxicity. So you don't need to have all this toxic effect of this drug in people who are already having leukemia. So you actually should lower the dose in about 10% of our population. And then the last one was tacrolimus, which is an anti-rejection medication. So all of your solid tumor transplant docs and your hemonc docs use tacrolimus to prevent rejection. And in our population, about 7% should have a dosage increase. We have two new things that are launching this month, CYP2D6 and another gene for warfarin. And then in January, we'll be launching HLA B5701. So let's look a little bit about this. Before we go into each of those, I want to talk a little bit about something I mentioned at the beginning, which was preemptive testing. So preemptive testing, when we launched the test in 2010, the clinicians could order it two different ways. They could actually order it because of a clinical need, so the person is taking Plavix right now, or they are taking simvastatin right now, so they could clinically order it, or we searched the EMR of our 120 or 130,000 patients who are kind of our home users. And you could read their ICD-9 codes and know that they were at risk of perhaps going on one of these drugs. So when they came in for their well doctor visit, there would be a little thing over here that said, click here to let us tell you about PREDICT, and it would tell you this person has a probability of taking clopidogrel, simvastatin, or warfarin in the next 3 years. So go ahead and order this test now. So how would you do that for billing purposes? Well, at this time, this was covered under a grant support. So we were able to do this preemptive testing on a large number of patients. So if any of them went and started taking clopidogrel or warfarin or simvastatin in the next couple of years, their data would already be stored, so the pharmacist or the clinician would know which drug to order and how much of it to order. So two different things were going on: our preemptive testing with data storage, as well as our clinical testing that was relevant to the patient at that time. So now it continues to evolve, PREDICT, and we have paperless orders now for PREDICT, where they list that that's the test they want. They have to choose which drug group they want. Like, are they interested in an antiplatelet medication? Are they interested in an antidepressive? Are they interested in a statin? So they will choose which drug that they're most interested in. These are all the different drug groups that they're given. These are the different genes, because they never remember which gene goes with which drug, nor I can hardly keep that straight. But that's all linked to different CPT codes. So they will order one drug group. We will then bill a specific CPT code that went with that drug group, so it's relevant to the patient. And then they will all be given one price, and all the results will be stored. So even if right now they may be interested in an antiplatelet, they will have the data for all these other ones in their EMR. Okay, so here's another example. So this would be CYP2C9, VCOR, or CYP4F2 for warfarin. So in this case, we would look at 4 different alleles for CYP2C9, one common allele, which is a promoter region in VCOR, and then another variant that's been found in CYP4F2. So we test all of these together, and together they would give you the correct dosage of warfarin to give to a patient. So if someone was a star 3 and a star 1 for CYP2C9, they had 2 variants, or they were homozygous for the promoter variant for VCOR, then it would say that they are a hyper-responder. Starting dose should be required. They have 2 VCOR and 1 CYP2C9, so you should change their warfarin. Again, there is no current evidence for pediatric warfarin dosage. Again, they would go into warfarin decision support, and they would put in various parameters about the patient, their age, their height, their weight. It would know also in here the genotype, and that would tell you the correct dose of warfarin to give. Now, this is the Vanderbilt calculator. There's another one out there that's I'm just blanking on it. Warfarin, anyway, you can go online. It's out of St. Louis, and they have a similar kind of things where you plug in information about your patient, and it tells you how much warfarin to dose. That particular link requires CYP4F2. So if a Vanderbilt provider didn't train at St. Louis or isn't familiar with CYP4F2 and wants to use their the St. Louis guidelines, they can supply the data that we've given them and use that one. If they want to use the Vanderbilt calculator, it only takes into account CYP2C9 and VCOR. CYP4F2 is not worked into this calculator. So anyway, we provide CYP4F2 for providers who may want to know the genotypes for all three of those drugs. Okay, again, we're not talking about insignificant numbers of people. We're talking about only about 45% of our patients who actually would have the normal dose based on the FDA guidelines for warfarin. Then you have both the poor metabolizers included in here, where you mostly have a dose reduction. So SLCO1B1, there is a particular variant, star 5, which is well reported in the literature as affecting the ability of this transporter molecule to bring simvastatin in. So this person is heterozygous for this variant. So this person would be called an intermediate myopathy risk. They have one low functioning transporter allele. And again, pediatrics is less well known, but we do tell you what the result was, and again, refer them to my drug genome. Here's the decision support for simvastatin, which basically says this person is an intermediate myopathy risk for simvastatin. So consider another statin. And it goes into detail here for the provider. Again, this is not rare. 74% of our population, we wouldn't change anything about the typical dose, but in 24%, we would say to lower it considerably, and in 2%, switch completely to a different statin. CYP3A5 is associated with tacrolimus, which is the anti-rejection one. In this case, the person is a star 1, star 3. Again, it tells them that. And here it says this person is a hypo responder, and I'll show you the interesting thing about tacrolimus and CYP3A5. You'll notice that the majority of our patients are star 3 alleles. That's because when the clinical trials were done, most of the patients were Caucasian. That actually is a poor metabolizing allele. So when they determined the correct dosages for tacrolimus, it was based on people who had poor performing or poor metabolizing alleles. So in the patient population, which is largely in the African American population, where they have the wild type or the normal allele, they actually need a higher dose of tacrolimus because they metabolize it so quickly that for this anti-rejection medication, you want a trough level or a constant level of the drug in the system in order to prevent rejection. But in a normal allele or in an African American who may be a star 1, star 1, they need a higher dose because they are metabolizing the drug so quickly that they're not maintaining that level within their system, and they are at risk for rejection. So this is a little backwards from most of the other ones that we tend to think about. And then there's TPMT, which is the methyltransferase. So you want to limit the amount of this very toxic drug to these patients. And so in this case, you take some of the drug and you inactivate it by methylation. Part of it's active and part of it is inactive. If you're a poor metabolizer and you can't inactivate a portion of the drug, then you have the active molecule, which is very toxic to the patients. So if you're a poor metabolizer, you don't need all that excess drug circulating. So reduce the doses to prevent toxicity to the patient. So this just shows a person is homozygous, a compound heterozygous for two poor metabolizing alleles. So there's an increased myelotoxicity, and so you need to lower the dosage. Here is the decision support associated with that. And again, about 10% of our patients should have a lower dose. Okay, so all of these results, all of these DGIs, these drug genome interactions, which are specific for a particular drug, we give you the genotype as well as what to do if you are taking a particular drug. So these DGIs are all in the EMR in prime real estate. They're on the front page of the person's EMR. So this is how they're displayed in the patient summary. And then every time I showed you decision support, I mentioned my drug genome. So I would refer you to this great source. I don't know if any of you do cancer genetics, but Vanderbilt launched the My Cancer Genome in 2011 maybe. It's a great resource. It was one of the first of its kind in terms of what does it mean to have a certain mutation in a tumor, and what does that mean to have a BRAF mutation or whatever. So based on that premise of My Cancer Genome, we launched My Drug Genome. And it's a wonderful resource. It tells about all these different drugs and all the different variants, and you can go learn about them. You can actually plug in different results and see it here as well, and the specific results. Now for CYP2D6, which is a little bit more complicated, there's many different types of drugs. So right now, all we offer is the metabolizing status. This person is has two poor metabolizers. We actually don't link it with a particular drug in the EMR. We are doing surveillance in order to figure out what is the most commonly prescribed drug group for which CYP2D6 is used, and then we will establish DGIs associated with those drug groups. But right now, you can go, the provider can go to My Drug Genome, and if they want information about coding, we can actually tell them what to do. So the DGI is here, it's just in My Drug Genome and not in the EMR. The other great reference is PharmGKB. So if you're interested in this topic of pharmacogenetics, they have more information than you probably want. It has all different pathways of this of these genes and their proteins and all the different pathways there, and it has all the different variants. There's so much information. That's another great resource. And this again is what I was telling you for CYP2D6. These are all the different variants that we test for, including a CNV. So we will provide the metabolizer status, but we don't have a specific drug associated yet listed, but you will be able to find that if you go to My Drug Genome. So the evolution of PREDICT. PREDICT has been around for 7 years. I think it's aged me about 20 years during that 7-year time. We have had what you would probably call the perfect storm during these 7 years. We've had to switch the format, the assay format, midstream from one assay that was then discontinued abruptly but it was a closed system, to TACMAN, which is an open system. And what that means, a closed system meant we couldn't add genes, we couldn't add variants, we didn't have to do the interpretation. It was closed in that way, and so it was nice that way. Well, nice and not nice. We couldn't add more genes, but we didn't have to interpret it. Now we have a TACMAN assay, which allows us to have an open assay. We can continue to add new variants all the time, but the analysis is much more complex. So we went from 6 genes and 16 variants to 8 genes and 31 variants and 1 CMV. Went from about 13 hours tech time, and that's not like 13 hours from start to finish, including lots of incubations. That's actually 13 hours of hands-on tech time for 30 samples to 196 well plate. And this one was no analysis, and now we are 5 hours, but we can only do 5 samples, 2 96 well plates, and a CMV plate, so 3 plates for just 5 patients. And it's a lot of hours of analysis. We went from getting lots of no calls and no results and only 98% accuracy to basically no repeats. The TACMAN assay is wonderful with greater than 99, I'd say 99.9% accuracy. 1 gene to 8 genes over time and associated DGIs. Our personnel turnover has been at least 70% during this time. We went from grant support to third-party billing, which created havoc with numbers and test orders and all of that. Of course, molecular CPT coding in 2010 was technical. Then it went to panel, and now we have all these genes, so we've had to change all the billing and CPT coding and all of that. We, of course, had a new lab informatics system in the middle of this where we went to GE to Cerner, which is like night and day. And now we have a hospital computer system that we're changing. So we went from our home brew, which has been around for years, Star Panel, to Epic, which is completely different. And you can see with all of this informatics support, this all kind of becomes very complicated when you have all these changes going on. So what are the outcomes of PREDICT? Well, first of all, the availability of pharmacogenetic testing to our patients, actually able to determine the percentage of our patients with drug-altering genotypes requiring dosage or alternative therapy. We have development of a model for simplified and compliant test ordering and billing. We've identified a method for storage and displaying genomic data for the preemptive testing. Many of our patients have a lot of this data just kind of sitting there waiting to be used. You know, they have CYP2C19 or warfarin, but they're not on it yet. So we haven't really been able to totally look at the great clinical utility of all of this. We're just kind of collecting lots of data right now. And we have been able to establish informatics tool for the automated DGIs and healthcare provider decision support. So some future of PREDICT is to, like I mentioned, to measure the clinical utility of the pharmacogenetics in routine patient care, hopefully looking for a reduction of ADRs and costs, expand throughout the VUMC network, because Vanderbilt has a main campus, but then we have lots of new satellite areas across the southeast, so be able to offer this throughout all of those. Continue to add new genes with associated DGIs, all of which is going to be evidence-based. Continue to educate our healthcare providers, because this is still really Greek to them, and create the availability of PREDICT with non-VUMC affiliated hospitals. And these are just some pretty pictures from our lots of our South American trips, but actually they all kind of can somehow link to predict. So when you think about this nice, big, beautiful waterfall at Iguazu Fall, I think of predict as we maybe have a little bit of knowledge and information of one of these little waterfalls. But once pharmacogenetics is totally understood, that it'll be just this mass of information, this mass waterfall. I think there's still a lot of mystery about pharmacogenetics, kind of like Machu Picchu and the mystery of the Incas when they were there. I think the variance in people and how they respond to different drugs could be as unique to all the different types of mountains that we have across the world. This is really cool. This is a desert scene. If you were to stand at this site throughout the day, you would have a totally different view. So you may have the same gene, but lots of different variants in it. And this, I couldn't really figure out with pharmacogenetics. He was just kind of cute. But I imagine it also helped balance the slide. But I imagine that drug metabolizing genes in the blue-footed booby is probably quite different than us. So anyway, I'll take any questions.
Thank you. That was that was an excellent presentation.
Great.
A question that I have for you, actually I have two. I think you partially answered one, and that was to how are you going to evaluate the benefit of this? I'm sure that this adds cost to patient therapy. So as far as a reduction in adverse drug reactions, drug cost, you know, what are your plans real briefly?
Right. So that's a big deal because that's really the main reason why we did predict was how can we reduce ADRs and show the cost effectiveness and clinical utility of this. And that unfortunately will take time because we've determined that our patient population, many of our patients would be candidates who should get different doses and different drugs, but it's going to take numbers to actually measure that. So we're just collecting data, and you know, as our patients start going on these different drugs, and we measure the decision support. Actually, I think that's interesting too. How many times do providers do what we suggest, or do they continue to do with what they know to do? So we've got lots of things to measure, and I think that'll just take time to get those numbers. But right now, it's all being collected.
Great. And my second question is that just two of the examples that you used, the one was clopidogrel or Plavix, the other was warfarin. Those drugs are also used in the outpatient setting.
Right.
So if patients are using your predict information, are switched to, let's say, the more expensive medications in the outpatient setting, have third-party payers been willing to accept the information from predict as far as paying for those more expensive medications?
That's a great question because you are correct. Simvastatin, for example, is one of the cheapest statins, which is why it's most commonly prescribed. Our data would support that the third-party payers are on board. You do need to educate them. So that's kind of the providers that, you know, we work with locally. They're kind of on board with this. But that's not to say that in other states or other providers that they may be as accepting of switching drugs like that. But then the provider becomes the advocate. And so make that happen.
Thank you.
Hi, just actually to address his first question, Paul Goebel is actually doing, you know, this work, actually evaluating this right now up at Inova.
Oh, okay.
So you might, I don't know if you know Paul or not, but he's actually using bedside genomics, you know, in ACS patients right at the time, and they get the results in 1 hour.
Wow.
And then they
We
figure
can't
out
promise
which drug
that.
to do. So it's a really interesting, it's the same technology, but they have a bedside unit. And so they're actually able to do that, and they're running a trial right now.
Wow.
I'm not sure what the name of the trial is, but
That would add more age to me, I'm afraid. Making sure that would be done right. Yes, okay.
But my question is do you think that, or maybe how soon, do you think that this kind of technology will be able to be incorporated into new drug applications to the FDA? Because it seems like we could resolve a lot of efficacy and safety issues at the same time and decrease the size of clinical trials by using this kind of information.
That's a great question, and I don't have the answer to that. But that's a good question. Hopefully, as more of this information comes out and these trials come out and they see the efficacy of knowing this up front, maybe that will be a component of clinical trials and the FDA requirements and all of that. But things in the government move a little bit slower. So, and if you're asking the FDA to be involved, you know, who knows where that's going to go.
Thank you.
One quick question. What's been the feedback from the clinicians who are using the various EMRs and get the alerts? Because one of the problems that we have in the clinic and systems like Epic and all that is there's a lot of alerts that come up, a lot of reminders. And I think it speaks to his question, how many times do clinicians just click through and say, I can't deal with this right now, you know, and go ahead and do what they were going to do anyway?
That's a great question. A lot of provider education is required. So we've really been hammering this hard so that our providers are familiar with it. There was some months when we were at our peak where we were getting 500 and 600 samples a month because of those alerts, getting this person to have testing done. That's also when it was free and covered by a grant. So when when third-party payers started, then that dropped, and our new system will, I think, be great too, because the person may be on a drug and they're going to get all of these other ones. So our providers love it as long as you continue to educate. That's really the key. Okay?
Thank you, Dr. Vnencak-Jones. Our next speaker